So I'm going to switch gears a little bit from the last presentation and try to just give a broad sort of 10,000 foot view of what we do at Larval and how we're trying to bridge sort of the information gap with all of the vast um, data coming out in predictive biomarkers and oncology, how we adapted our technologies and how we've been building a new platform and modifying our approaches that we've built over the past 15 years in the competitive intelligence space, and working to build new tools that are specific for predictive biomarkers in oncology. So perhaps a bit of a probing title, why is monitoring predictive biomarker data challenging for pharma? I think the shortest answer could probably just be described as the sheer breadth and depth and how to escalate that quickly, which is sort of the space that is competitive intelligence. So just going to give a broad overview of the direction we've been heading. Ovarian biomarker, predictive biomarker, competitive intelligence is our newest area. So really honored to be here, be surrounded with such incredible researchers, scientists in the room. And I'll show you our platform as well for your exploration and feedback, and looking forward to maybe connecting through our virtual lobby area. So to give just very broadly an introduction to Larval, for the past 15 years, we've been really in the business of, in the software as a service, of using natural language processing and text mining tools to bring competitive intelligence to major pharma companies. A couple of our current customers, partners here listed on the slide. So traditionally, it's been really driven by completely automated text mining and natural language processing, being able to bring news items ranging from PubMed to market updates to FDA approvals, and getting those as a pulsed alert or a pulsed email or an indication to our customers, and focusing mostly traditionally building around specific drugs, specific MOAs, or specific disease areas. And we've also been working on automating and indexing conference abstracts into conference planners. So that's something we've been working on for a number of years. When we sort of got inspired and really motivated to move into the predictive biomarker space, we sort of came to see that we needed to customize our approaches, add new layers, and really put the context needed on this vast amount of data. And we're currently covering a large number of both solid and hematological malignancies. So that's a bit of the background of what Larval does. And in addition, we also do KOL monitoring. So we use similar tools to be able to track key opinion leaders in the field. So when we first started to endeavor last year, when I began building a team out, we started to look for some inspiration from some of the other academic and commercial groups who have sought to do databasing of cancer biomarkers. And I think with the attendees in this meeting, you're probably already well aware of some of these. There are a number of great free and academic driven biomarker databases, one of which is Civic, which has been able to build a breadth and depth by doing sort of the crowdsourced model, that they have an open sort of access, the ability to submit particular genes as long as it fits within their hierarchical structure, and they are approved and then go into the Civic database. And it has really great breadth, but it utilizes that sort of crowdsourced model. Also a little bit maybe near to my heart, I did my postdoctoral research at Vanderbilt, and there's also a project called My Cancer Genome at Vanderbilt, which focuses more in on bringing together all of the trial information to sort of put it to researchers and I guess somewhat patient focused as well, identifying trials that meet their profile. So there was a lot of, we thought we could sort of fuse what we do in pushing competitive intelligence, daily alerts, pulse emails, and integrate it with the approaches of these academic sites And we started to be inspired that way to start building Very, which was mentioned as the platform that we're building now. And of course, with this audience, you're probably also already well aware of some of the commercial resources for predictive biomarker in cancer. Uncle KB clearly has a really good breadth and depth, and also by being commercial, they had a higher level of curation, meaning not just having sort of the data in flat form, but also being more curated, summarized, and annotated, having sort of a higher threshold to move through to get into the database. And also Jackson Labs has a product called CKB, which has a really, as one might expect from Jackson Labs, a really good depth of data within the preclinical space. So we were really inspired looking at some of these other tools in the marketplace, and we're beginning to see that we needed to complement what we do with natural language processing and pushing of news, and being able to collate, curate, and index that information in such a way that it can be of greater utility for our customer base and our market. So this is when we started sort of assessing what our tools could do, but what our tools needed to be built out further to accomplish. And what you're seeing here, I hope it's not too fuzzy on the slide, this is what we can do with our natural language processing. We can call out per product all of these news items ranging from Globe Newswire to updates in ct.gov or UserCT. And we're able to pull out with that NLP all of the types of data, trial status, obviously the tumor types and the products themselves. And we started here and began to build all of the text mining expression to pull out all of the specific biomarker genes and alterations. So we're covering now hundreds of genes and in the thousands of specific alterations, be those point mutations, expression level changes. We've built the language tools to pull that out in the automated news. But as one could probably surmise, this is a bit overwhelming. It's sort of a bit of a fire hose of information. Even if you just look at one indication, such as breast, it's just a large amount of information coming through in any given period. So it further inspired us to want to build on top of our text mining tools and be able to curate the data and build a team to put that context in the data, pre-screen, and push that from a CI perspective. So this is a bit of a look of what that is. This next slide is intentionally zoomed out and difficult to read, because one of the other major things we want to bring with our softwares is alerting to major conference abstracts. So we've already built the tools to automatically index conferences and put them into planners. But in terms of biomarker data, the sheer density breadth of it, here this is a visual actually summarizing in the center the types of tumors, in the next loop the types of genetic alterations, and in the last loop the specific drugs. This is just what came out of ESMO recently, and obviously it's a huge amount of information to digest, and that further is, in terms of what we do with competitive intelligence from all news, trial updates, and keeping our market abreast of conference highlights, realized we needed to build a team to add to our tools. So one of the main ways we wanted to do this, and again, reemphasizing the point, how do you put together and organize all the contextual data over time? Here, just looking at a visual, an NSCLC, this is what we're covering in terms of the number of alterations we've identified in our database. How do you put all this information together? Here, just for lung cancer, non-small itself is a bit overwhelming. So where we started is to try to build a heat map approach to create a visualization of the data. So this is a bit hard to see, so I'll introduce it verbally, and if my screen share will permit me, I'll actually maybe pull the platform up after that. 1 second. So what we've done and been able to organize by disease, by drug, by gene, by mechanism of action is a heat map. So what we've done is engaged in a manual curation approach assessing all of our news inputs for specific relationships between the biomarker on the left, the targeted therapy on the top, and obviously the tumor type, and curated and tagged all of that based on AMP-cap levels of evidence. So we're compliant with that system, as are a number of other tools in this space. So we code all the way from the highest level of evidence, according to AMP-cap, which is obviously a regulatory approval, which is on the A1 level, and we code all the way down through B, late trials, and down through early case studies, as well as preclinical data. And in the visualization, obviously, the relationship between a drug, a biomarker, and an outcome is something that's not flat. It's something that progresses over time and also has, there's more context outside of just those 3 points of information. So what we're doing is stacking the data on top of each other. And what you see on the right here is a stack of the relationship between a particular biomarker, a drug, and a cancer, but there are different levels of evidence within that one relationship. So we've built these three-dimensional visualization tools for heat maps to put all of that data together to get quick eyes on and link it all out to the source to be explored from there. So we're very, it's really, I think, would probably be self-evident to those on this call. This is very much a first level of the rocket. That we're just trying to get a pivot point to get all of this breadth of information in a digestible form to our customers, as well as for research purposes. So I'm actually going to, so it might be a little bit easier to see, pull up our platform to just do a quick visual of what we do. And I want to preface this by saying I'm not a salesperson myself. In fact, veri.larval.com is completely free for research non-commercial purposes. And given the amazing talent on this call, it would be wonderful if you wanted to check it out and provide us some feedback. This is what we're building, and this is our free platform. So what we've been able to do is structure all of this data around cancer types, around particular biomarkers or biomarker genes, drugs, drug classes. And we've also been able to build around companion diagnostics and other clinical diagnostics as well. So just to sort of demo what we're venturing toward with this new Veri product, which has only been launched since ASCO, quite clearly we can put together, using our NLP and text mining tools, we can pull all the news coming out of trial updates, PubMed, et cetera, put them all in one space for you and annotate them by all of their particulars. Herein, we've got an MSI high DMMR as a biomarker related with Keytruda. We can put all of this data together from the news, also from social media, and as well as pulling all the trials together that fit the parameters of your search topic. If you're on non-small cell lung cancer, you can pull everything out there. And here, maybe it's a little bit easier to read on a larger screen. If I go over here, and I'm getting a bit of freeze in my Zoom, if you'll give me 1 second. I need to stop share and let me re-share. Sorry about that, everyone. Technical difficulties. So this is a view of the ground we've been able to cover in non-small cell lung cancer. So obviously, even when you put it together in three-dimensional space, it still can be a bit overwhelming looking at all of the biomarkers down the left-hand side versus all the targeted therapies on top. And so again, all of this has been manually curated and assessed. We've actually realized that at the first level of things, while there's absolutely lots of opportunities for applying NLP to this type of public information to bring it together, we've really seen that the first driver of this that needed to have a manual assessment. So we built up a team of master's levels and PhDs and MDs who are able to process this data on a daily basis and both push it as news and as well as put it into this three-dimensional visualization. And again, this is a free platform. If anyone would love to play around with it, use it for non-commercial use, would love some feedback or just to see if it could be helpful to you. And to finish, I guess to finish my point on this, we also realize it's critical to attach some of this evidence that we're pulling, annotating, and collating, and try to affix it to the companion diagnostics and diagnostic tests as well. So as you can see, we're trying to keep an active track of both what's already stable, and I'm sure all of you are well aware of these approved companion diagnostics and some of these indications, but working to link together all of that data as well. So pulling in that, we're tracking around 220 tests in the news, putting that together with our evidence, be it ranging from approvals to when these branded diagnostics are being used in earlier work. Obviously, I happen to be on the Roche page, so they have a lot of approved diagnostics, but we also capture it when it's being applied in a late trial, just to give a view of how that's going in the market space, as well as building the natural language processing, my favorite word of today, to pull the news together as well. So tracking 200 plus tests in around 50 companies, getting all this information into one platform is the governing goal of what we're trying to do at Verity. So again, very much a 10,000 foot overview, and really just hoping we can use our technology to sort of bridge that informational gap to restate it. There's just so much data out there in the universe. How do we put this all together and put into a digestible format for it to be actionable? So first at a pharma competitive intelligence, speed of knowledge basis, but there's a lot of other places we hope to take that in the future as well. So to set that up, and I won't have as much of a detailed visual here, but we're trying to build on this foundation and apply this publicly available data and think of new ways to build upon it, new ways to make it useful for different audiences, be it research, be it clinicians, maybe other areas in the healthcare space. So some of the things we're working on right now is applying sort of a artificial intelligence approach, which given the some of the individuals in this room, probably try not to speak in too much detail on AI, but we have an AI team based out of France who is building out some approaches there. We're also looking at doing more trial tracking, both from sentiment analysis and such, as well as patient profiling. So to just sort of make quick comments about some of the things we're working on at LARVOL in this space. Within AI, you may have noticed when I was demonstrating the heat map, as is probably obvious, there's a lot of white space. So what we're looking to do is take the flat data that we have, where we have the tumor types and subtumor types, the interventions, the mechanisms of action of the interventions, the genes and the specific types of biomarkers. We're looking to apply a TensorFlow to that full data set to see if we can come up with a strong predictor of where new areas of research may be merited. And so that's a recent effort, and looking to integrate that predicted data with the empirical data, as you might say, which is what we're focused on showing on the front end right now with Veri. So very exciting venture. And as someone who spent 10 years at a bench trying to validate 3 or 4 biomarkers, it's quite fascinating what you can do with enough computational power to predict new areas to pursue. So that's what we're approaching there. Something else we're working on as well is there's just so much to keep up with in terms of trials that we're looking to actually recalculate our data in a different context, which is putting all of the relevant public data, even tracking over the full history of a trial, and putting all that data together under the one hat of the trial's name. So giving you an access point and a searchable access point by trial and seeing all of the data as well for your own research or commercial purposes. So that's definitely one effort that is a restructuring of the back end that we've been building. And we're also very soon, if you were to explore Veri, you might see some Twitter tags on there. And that's integrating our skill set from our KOL tracking to actually do some sentiment analysis on trials and other topics in the biomarker and CDX space. So we'll be able to tag our news and some of our evidence with some of that social media sentiment analysis as well. That's definitely somewhere we're taking it in the future. And finally, I think I've used the phrase earlier in the presentation, the first level of the rocket. And I think very much with Very platform, it is the sort of foundation and that first level of the rocket. And we're really looking to add more sophistication to the data to bring it to other audiences. So right now, Very is really very much directed for the research side and for the pharmaceutical competitive intelligence space. But we really want to see what we can do to push closer to being physician facing and how to be a tool to help practicing oncologists as well. So one thing we're doing is pulling out additional information through manual curation, actually, and breaking down the relationships that I noted earlier between a drug, a biomarker, and a tumor, and pulling out what all of you are probably already thinking. It's so much more complex than that. So we want to pull out the relationships and predictive sensitivity and versus resistance and put them in the context of the stage of the tumor, the line of therapy, where they're appropriate, so that instead of searching by topic, you can search at the level of a patient profile. And the data can basically be sort of reverse engineered to give you the product that's related to all of the in the particulars of a patient profile. So that's another effort that we're working, and just really excited to be here at the conference and have the opportunity to give sort of the 10,000-foot view of some of the things that we're working on at LARVOL. So actually, I think I might be just a minute under time, but wanted to pause here to obviously say, feel free to reach out to me or any of my colleagues on the wonderful portal that's been built for us here. So if you'd like to reach out to connect with me there. And again, if you're interested in checking out Very for free, just go to veri.larval.com. If you wanted to go check around for your favorite biomarker of interest, hopefully we have good coverage there. So I will pause.
Excellent.